Thank you, Haishia, for a very generous praise. I wish I measure up to what you've told, so let me try to get there. Am I audible to the people at the back? Everybody? Okay, good, thank you. All right, so I'm going to kind of span, there's a lot of early career researchers here, so I'm going to kind of give them a sense of how to develop a research question and a story and develop a grant and science. And at the same time, I also know there's a lot of clinicians in the group. So I'm also going to touch some clinical points that I would use for my own teaching, training residents, et cetera, et cetera. So I'm going to kind of go with that. So I wanted to begin by thanking Martin, Jonathan Savitz, and Haishia for inviting me, Kent, Bart. They're all friends of mine for a long time. It's a pleasure. It almost looks like I've been in Tulsa many times before, maybe in a previous incarnation. But anyway, so I'm here. And I do think that I also wanted to mention a word of thanks to the William K. Warren family for supporting. I mean, this is a really profound gift that they've given to the society, because the kind of research that you guys are putting out here is going to benefit a lot of people, not just Tulsa, frankly. So I really wanted to genuinely convey my appreciation to that kind of gift. I mean, that enables people like us to do what we do. So without much ado, I don't have a lot of disclosure, so I'm going to just get going. So I'm going to start with trying to explain why should anybody care about inflammation in mood disorders. Now, inflammation is kind of considered a non-specific signal. CRP goes up in every kind of medical disorder, so why care? Secondly, I'm going to talk about why care about glutamate dysregulation in mood disorders. This is kind of self-obvious now with the development of ketamine and other agents. And thirdly, how does the glial dysfunction that is related to peripheral inflammation link glutamate, inflammation, and depression? Finally, I'm going to talk about how inflammation, glutamate, and all of these dysfunctions, inflammation, glutamate, and glial dysfunction lead to mood regulation, dysregulation. How does it dysregulate the brain circuits that control mood, leading to depression? And then I'm going to talk about what are the other mediators? Because, one thing I want to start by telling you that immune molecules don't have a ready access to the brain. They have to use a lot of mechanisms to get to the brain. So how does peripheral immunity, how does it even communicate with the brain? So I'm going to talk about Chi Neurons. A lot of inspiration has come from Jonathan Savitz's work here. He's been doing this for a long time. I'm simply going to extend some of his findings. And finally, what are the putative emerging targets for treatment, okay? So let me begin with some introduction. Depression is a disorder that's very common, impacting about almost 20 million adults in the US, about 40,000 suicides per year, probably more because suicides are generally underreported. It's the leading cause. It's probably the 10th lead. It's one of the most leading cause of disability in worldwide, according to the World Health Organization. The tragedy is about a third of patients who are treated with depression, and I apologize, not medications, also like CBT and other forms of therapy, don't really achieve full control of their symptoms. So thus you're leaving behind a large gap of patients who are not responsive to any kinds of treatments. Thus there is a need for newer conceptual frameworks and targets that would help us improve treatment for these patients who don't respond to anything. So one of the targets, I'm going to try to convince you that one of the targets is inflammation. There are many other targets. I'm going to be respectful of the field at large. So let's begin with what are the evidences. I'm going to summarize this in one slide, but this is a large volume of data. What is the evidence supporting that inflammation plays a role in depression? Now, let me stay at the outset. Depression is not an inflammatory disorder. Let me be very clear. Not all depressions are inflammatory disorders. MS is an inflammatory disorder, not depression. Some depressions does have some inflammatory activation in them. And that's what people like me and Jonathan study. So this is basically, we are kind of focusing on this subgroup, but that's not a small, it's about 1/3 of all of these depressed patients. So there is a fairly large group that we're talking about here. So some depressed patients have been, about 5 meta-analyses have confirmed that at least a subgroup of patients, about 1/3 of them, manifest all signs of inflammation, including elevated cytokines, which are inflammatory signaling molecules, such as IL-6 or interleukin-6 and TNF alpha. They also there's significant data supporting elevation of CRP in depression, and Haishia and Jonathan have recently published on that as well. And chemokines, which are basically these chemical signaling molecules that attracts the monocytes to the site of infection and inflammation. So all these are elevated in patients with depression in the absence of any kind of medical cause to explain them, meaning there's no infection, there's no diathesis that causes this. Intriguingly, early inflammation predicts later depression. Like people who are not depressed, and you study them very earlier on in their lives, people with the high levels of CRP tend to have higher risk of developing depression later on in their life. These are really huge, large-scale follow-up cohorts in the UK. It's called Whitehall 2 cohort. There is a Danish cohort. There is a Rotterdam, Netherlands study. So all of these cohorts have kind of confirmed that people with the elevated inflammation earlier on are at risk of developing depression later. Inflammation predicts decreased response to treatment, and we have published, our group has published, Jonathan has published on this, so a lot of people have published on this. There's a lot of data that depressed patients with the elevated inflammation have poor response to medications. Inflammatory molecules and inflammatory stimulation, like for instance, you can administer endotoxin, which is part of the bacterial cell wall of gram-negative bacteria. And that triggers an inflammatory response once you administer that for a short term, and that can cause symptoms of depression. Interferon alpha, which I will be talking about fairly extensively, can cause depression. It's an immune cytokine that's used for the treatment of cancers, and it's used for hepatitis C. And finally, depressive symptoms following cytokine antagonist therapies. And we used infliximab, which is a TNF alpha blocker, to show that it can have an antidepressant effect. Jonathan has used NSAIDs, aspirin, and so there's a lot of data out there. It's just bubbling. It's not completely fully formed, but there is a good nascent data set that's supporting that. Now, I'm going to quickly course through this. A lot of people know this, but I'm going to give you a quick 2-second overview on glutamate. Glutamate is an excitatory neurotransmitter. Now, when I say glutamate, it's like oxygen to the brain. It's like 80 to 90% of brain work gets done by glutamate. So it's almost so ubiquitous in the brain. And about 80% of the energy consumption of any activity in the brain results, it's due to glutamate. Thus, glutamate increases explain findings like PET FDG uptake scans, bold fMRI effects. These are all glutamate-related effects. Glutamate is co-expressed and regulates other neurotransmitters. The classic example is dopamine. Presynaptic dopamine receptors have glutamate terminals on them that regulates dopamine activity. There's also GABA neurons have NMDA inputs on them. And glutamate is a precursor to other molecules, including GABA, glutathione, which is an antioxidant. I'll explain that in a minute. And it's consistently elevated in bipolar depression. In fact, it's one of the most consistent finding in bipolar depression is an elevation of glutamate. There's multiple meta-analyses that have proved that as well. And it's also elevated in some unipolar, but not all unipolar depressions. It's implicated in all aspects of mood disorder pathology, which we'll see in a few minutes. Now, let me give you a quick overview of the glutamate cycling and dysregulation. As you can see, this is a tripartite synapse, and this is the presynaptic neuron, and this is the postsynaptic neuron. And the purple structure is this astrocytic membrane. It kind of encircles, as you can see, it encircles and cuffs the synapse. And this cuffing is extremely important because it protects the glutamate from escaping into the nearby environment. Because why? Because glutamate is toxic. So now let me begin with the question, where does this glutamate come from? It comes from two sources. One is it comes from metabolism. Tricarboxylic acid, energy cycle, Krebs citric acid cycle can produce glutamate, but it's a very small amount. But most of it comes from a precursor known as glutamine, or GLN here. And so the glutamine is floating around in the extrasynaptic space. The neurons uptake this glutamine, convert them into glutamate, and the glutamate is then carefully sequestered in these vesicles. These are known as V-glut, or vesicular glutamate transporters. These are glutamate vesicles, and kept tightly protected. Why? If you leave glutamate alone to wander freely, it will cause calcium intravasation and cause neuronal death. So it's pretty toxic by itself. So the goal is to prevent it from escaping. And then the glutamate is released into the intrasynaptic cleft. And once in the synapse, it has three different methods. One is it can bind to the receptors and cause an effect, it causes the neural effects, causes neurotransmission, or it can spill over. A small amount of glutamate spills over into the extrasynaptic space. This is going to regulate what is known as tonic activation, meaning there is a chronic activation of these neurons. And I'll explain that in a few minutes. But the rest of the glutamate, almost 80% of them are reabsorbed by this purple membrane, which is the astrocytes, astroglial cells, and they are converted back into glutamine and they are recycled. Now, this is an important cycle because the integrity of the cycle is responsible for neural functioning and energy metabolism. Now, what happens in inflammation? Well, during inflammatory context, there is probably an increased synthesis of glutamate, which is borne out in many kind of neurological and psychiatric disorders. There's probably an increase in the release of glutamate as well, because when you do the spike potentials, they show up high levels of calcium calcium activity in the postsynaptic neurons. It also causes what is known as a postsynaptic potentiation. So many of these other things that are happening. In addition, inflammation kind of causes this other big problem where it suppresses the function of the excitatory amino acid transporters, meaning these are the channels that enable astrocytes to absorb glutamate. Remember, 80% of the glutamate is going to be metabolized by these channels. Inflammation, by impairing them, increases the amount of glutamate in the intrasynaptic regions. By increasing this, so as a consequence, there is an enormous amount of glutamate that's getting built up in the synapse, leading to potential toxicity. And as you can see, this glutamate tends to spill over into the extrasynaptic space. So in other words, during inflammation, the neurons tend to leak a lot of glutamate. The synapses leak glutamate. This excess glutamate is also joined in by glutamate that is released by these immune cells. There are many mechanisms. I can answer that in the Q&A. But nevertheless, there's a lot of glutamate floating around in the extrasynaptic space. And so what happens? This extrasynaptic glutamate is also joined by glutamate-like molecules called quinolinic acid. Jonathan has done a lot of work on this. And so that molecule also behaves like glutamate. In fact, it's actually a little bit even more potent than glutamate in some ways. So it does the same thing more aggressively. So all of these molecules tend to converge on the glutamate receptors. Now, you can see there are two types of glutamate receptors, one in the synapse and one outside the synapse. So this is intrasynaptic and this is extrasynaptic. The intrasynaptic glutamate promotes synaptogenesis. It builds synapses. It helps neurons connect with each other and form new connections. The extrasynapse actually blocks it, meaning it's actually toxic. It's going to suppress synaptogenesis. And so by engaging this extrasynaptic, the excess glutamate in the extracellular space engaging these synapses, blocks them and causes synaptic degeneration and synaptic loss. In addition, some of these glutamate also diffuses and connects with this presynaptic glutamate receptor, which is like an autoreceptor. It blocks the excess glutamate release. That causes decreased stimulation from the synapse. So there are many things happening that is going to cause synaptic loss, basically. Now, how do we study this in humans? Glutamate is a notoriously difficult substance to study because it's so ubiquitous. It's present so much in the brain. So we use chemical spectrographic measures. And we use that in the MR scanner. It's called MR spectroscopy. As you can see, I cannot go into the details of MR spectroscopy. I can talk to you one-on-one later. It's a full science by itself. But nevertheless, briefly, we take these chunks of brain tissue. These are not anatomically precise. Remember, these are just volumes of interest. They're not anatomically defined regions. And we take these volumes of interest and measure these glutamate signals. And the output, the chemical output from this spectrometric reading is like any other spectrometer or spectrograph readings. As you can see, it's a bunch. It almost looks like an EKG. Like there are some peaks and there are some valleys. The only difference here is the area under the curve of these peaks are reflective of underlying concentration of these metabolites. We're going to first focus on glutamate, okay? Now, let me get to the clinical side of things. So let me begin with the immune stimulation. Now, I'm going to start with interferon alpha. Now, interferon alpha, I don't know, many of the senior clinicians in the audience will know interferon a little bit more. You might remember that it's a pro-inflammatory cytokine that causes increased inflammation. And by promoting this, it's a type 1 interferon actually. So by promoting inflammation, it kills the viruses. It destroys virus and virus particles. So it's an indirect treatment for hepatitis C and other chronic viral infections. Unfortunately, has so many side effects, it's been superseded by other drugs. It's usually administered by the patients, self-administered. The patients administer them in their own thighs. Each dose, they give the dose at say 150 microgram or something. They give it once a week. So patients are taught to self-administer. They go home with 4 doses. Then they come back in a month. So as you can see, one of the major side effects of this interferon alpha is that it stimulates inflammation. We know that because it's a cytokine. But one of the unintended consequences that was noticed was it causes severe depression. As you can see, in 12 weeks, about 50% of the patients remain severely depressed. When I say depressed, I'm talking severe depression. So it's a very reliable inducer of depression. Is it a reliable inducer of brain inflammation? Because you're giving interferon alpha outside. You're giving it for treatment of cancer or hepatitis C. So this is a study that Chuck Risson and Andy Miller did, basically. So they measured interferon alpha and other immune markers in the brain after 12 weeks of interferon. And as you can see, the IL-6, MCP1, which is a chemokine, which is one of those, it's also called CCL2. All of these are substantially increased following 12-week treatment with interferons. These receptors are driven by the primary. These are floating receptors that neutralize the inflammatory cytokines, meaning STNF-R2 and IL-6R. IL-6R neutralizes IL-6 and STNF-R2 neutralizes tumor necrosis factor. So in other words, the cytokines are seen in the plasma. They're floating in the plasma, but they also have their own floating receptors. Why is this? It may be because the body is regulating, protecting you from too much of inflammation. There's some kind of internal control for this. Okay, now, what does it do? Let's take a look at brain imaging. What does it do to the brain? What happens when you induce inflammation? Now, on the left-hand side, this is a cross-sectional study after 12 weeks of interferon alpha. This is more like a location detection task. The patient's asked to press a button, depending upon which quadrant this yellow dot is in, and then depending upon the correct or the wrong responses, you kind of take all the correct responses and see the brain activation in that. As you can see, the interesting finding from this is that the patients who were given interferon alpha performed equally well. They made as many errors or correct responses as patients who were not given interferon alpha, but the patients who were developing acute inflammation from interferon alpha developed significant recruitment of additional regions. In other words, to achieve the same result, they were activating more brain regions. In some ways, there was some kind of processing inefficiency that you can see where additional regions were recruited. This location of this region is in the dorsal anterior cingulate cortex, which is kind of one of those regions in the brain that detects cognitive conflicts. So in other words, they're not able to process between the right and wrong stimulus, and in the effort to process, they activate more brain regions. The second one is a FDG PET uptake study. This is a radioactively labeled glucose. You inject this, and it kind of is uptaken by the active regions of the brain. And as you can see, after 4 weeks of interferon alpha, patients, there was an increased uptake of glucose, radioactively labeled glucose, in the basal ganglia regions. And this excess uptake of glucose, was associated with the fatigue and with lack of motivation symptomatically. Okay? So this is a pre-post study. Now, so we wanted to extend this finding. So we basically took a bunch of patients with hepatitis C who are about to start interferon alpha. We took 17 patients who are getting interferon alpha and 14 patients who are controls. And then we studied them at baseline and after 4 weeks with identical behavioral MRS scans and blood sampling measures. So what did we find? The first question was, does increased inflammation increase glutamate in the two regions that had already been shown to be altered in inflammation, namely basal ganglia and ACC. So the first question was, that's what we asked. And the answer is yes. After 4 weeks of interferon alpha, the glutamate in the ACC regions and in the left basal ganglia regions were significantly increased in patients who are taking interferon alpha. And the patients who are not taking interferon alpha did not show this increase. Okay, and we did not find a similar finding in the right basal ganglia. I can explain, there are many explanations for this. But nevertheless, the increased glutamate in the ACC region was associated with greater severity of depression ratings. The IDSC is a depression scale, and so the increase was associated with, this is a pre-post study, so it's a prospective study. So it was increased it was correlated with, or it was associated with depression severity, whereas the increase in the left basal ganglia glutamate was associated with the decreased activity levels. Now, this decreased activity is a scale. It's called multi-dimensional fatigue inventory, and in that scale, you measure the patient says, I'm not active enough, I'm not able to get up and walk. It's kind of these kind of self ratings of the level of activity, physical activity, which is somewhat similar to psychomotor retardation that we see in depressed patients. So the next question we asked ourselves was, is there a dose-response effect? Meaning, we have already shown that inflammation increases glutamate, but is there a dose response? Does it mean more? Is there an effect that the more inflammation means, does it mean more inflammation means more glutamate? I mean, that's a good question to ask. And for that, we had to stratify the groups into high and lower risk for inflammation. And one of the ways of doing that was aging. Now, this is an interesting study, and I have to tell you, so this is a microglial cell, right? So what happens is, normally the microglia look like this at a resting state. They're basically fibria. They have this extended arms called fibria. They're surveilling the environment, sampling what's going on around them. But once they detect an inflammation or a dangerous, what we call as a pathological molecule, danger-associated molecular pattern, they switch into activation mode, and they become like this. They become amoeboid and start moving towards the site of danger. But after completing the destruction of the dangerous molecule or the pathogen, they get back to this resting state. So what happens is they start here, they go to activation, and then they get back to rest. But as people grow older, they don't get back to resting state. They go back to this partially activated state, and it's called priming, where it's neither completely at rest or completely stimulated, somewhere in the middle. Okay? And this may be a protective mechanism. This is a consequence of aging at one level. It's also seen in other pathological states like repeated concussion that Jonathan studies. So it's also seen in other progressive neurological states. And the other thing is, as you can see, as we age, we use hippocampal volume about 60 years. After between 60 to 100, you're simply dropping off all the hippocampal, you're dropping off almost half your hippocampal neurons. And here you can see the same, it's a language task, and the old people, the older individuals, when they're performing the same task, they activate more of their brain regions. You see how much they're activating to do the same task compared to younger individuals. Even though the performance is equal, there's no difference in the quality of performance. It's just that they activate more brain regions. And this is somewhat similar to inflammation, if you may remember, where they activated more regions to accomplish the same result. So we took the same sample that we studied before, but we divided the sample interferon subjects into above and below 55, which is the cutoff, which was a median age. And then we compared the glutamate response as we had done before. And so, as you can see, the first thing is people who are older than 55 had almost twice the amount of increase in the immune marker. This is the soluble tumor necrosis factor receptor, which increases in response to the increase of another cytokine, which is Tnf Alpha. When that goes up, this also goes up. And so this increase, you can see it almost doubles in the quantity, compared to individuals who did not get interferon alpha. So after interferon alpha, this substantially increases, and this is comparably increased compared to other groups. But more intriguingly, did it we also noted that this was associated with a parallel increase in left basal ganglia glutamate. So not only did the inflammation increase, but the glutamate also increased along with that. And there may have been some kind of a dose response effect because one group was responding more to the inflammatory stimulus than the other group, the younger groups. And this increase in glutamate was associated with, this is again Tnf alpha, which is the cytokine that we were talking about a few minutes ago. But this correlation was seen only in the older subjects, not in the younger subjects. And it was associated with the decreased motivation, which is one of the classic symptoms of depression, and choice movement, prolonged choice movement time, which is, it's a reaction time task. You have to press a button. The duration between the task is flashed and the button pressing is the latency, and it increases the latency. So you can see that the data is indicating that we are tapping into several symptoms, at least some key symptoms of depression, namely motivation and psychomotor slowing. So now I'm going to switch gears here and change this to a depression. And as you can see, in depression, so we have so far proven that if you give them interferon alpha, they develop depression-like symptoms, which in turn is associated with the increase in inflammation and glutamate. But how does this compare to depression? Why? Because interferon alpha is a severe stimulus. It's like a jackhammer for inflammation, meaning it's almost like hitting something with a jackhammer. You trigger a huge inflammatory response. Whereas in depression, the inflammation is very subtle. It's kind of low grade. And in addition, in interferon alpha, it's acute. You give it, they become inflamed. Whereas in depression, it's chronic. It goes on for years and years and years. So in order to study this, we took 50. So there are some differences. So we wanted to test if these findings are applicable to depressed patients. And as you can see, we divided them into 3 groups: high, medium, and low inflammation. And this is based on a classic cutoff from CDC and American Heart Association, American College of Cardiology. And as you can see, we also hypothesized that there will be more glutamate in this high inflammation group, and that the increase in glutamate, or in other words, glutamate will have a linear association with CRP in this depressed group. So these are medically stable patients. Remember, all our studies have to be, the patients cannot be on medications. That's the main problem here. We cannot take patients who are treated with antidepressants because antidepressants themselves change the MRS findings. So we had to have medication-free patients, which is where the difficulty comes in recruitment. So this is a classic. As you can see, these are 2 voxels. One is anterior cingulate, one is left basal ganglia. We didn't do the right basal because we didn't have a finding in the past. And as you can see, the high CRP group had a greater concentration, or at least had greater measures of left basal ganglia glutamate in the left basal ganglia. This was seen in a group-wise and in a linear analysis. As you can see, more CRP meant more glutamate. So there was some kind of a relationship between these two. And this increase in the left basal ganglia glutamate was associated with what symptoms that we would usually associate with the depression and basal ganglia dysfunction in depression, like anhedonia. This is an Anhedonia Scale. So it was associated with greater anhedonia. It was associated with the slower finger tapping speed. It's a test of psychomotor slowing. This used in Parkinson's, where they ask them to tap, and the speed is measured. Reaction time, where again where you respond to a stimulus, and psychomotor speed of information processing. Here you give this digit symbol substitution test on the Wechsler-Arendt scale. So again, to summarize, the increase in glutamate associated with inflammation was associated with significant cognitive and motivational and anhedonic difficulties that are part of depression. Now, one thing we have to understand is that what I've told you so far is a selected region, one region of the brain and one metabolite, but the brain has more regions and more metabolites. So in order to do this, we use what is known as a multivoxel spectroscopic imaging. This here, you take an entire slab of a brain tissue, say for instance at this level, and then divide the entire region into 1 into 1 into 1 cubic millimeter voxels. Each of them are small voxels. And then you extract the spectrum, metabolite spectrum from each of these voxels. And then you can study different regions. For instance, we chose this region of putamen. You can see that, right? This putamen region. We focused on that, but other people focus on other regions. And in addition, as you can see, there are several chemicals that we studied, and one of them is called inositol. which is going to be very important for us because inositol is generally believed to be an MRS marker of astrocytic dysfunction. It's a sugar, it's an osmolytic sugar, it accumulates in the astrocytes, and when it builds up, the astrocyte is in a distressed state. That's when it starts building up. There is another chemical called choline. Choline is basically when there is a lot of cell wall synthesis going on inside the brain. It simply reflects there's more turnover of the cell wall, which usually happens when there is inflammation. And the third is NA or N-acetyl aspartate, which is a neural vitality marker. Basically, it's found only in the neurons. It reflects in mitochondrial metabolism of the neurons, basically. Okay? So the first question was, this is a completely different technique. Is it still showing the same findings we found with the other study, other MRS technique? As you can see, the high CRP again had high levels of glutamate in this study, significantly elevated levels. And intriguingly here, what we did was we compared the immune markers with some of these other markers, the glial markers that I was talking to you, especially the astroglial markers. And as you can see, the CSF CRP, I mean, we had spinal taps on these patients. So we did a CSF CRP, and as you can see, the CSF CRP was significantly associated with this astroglial distress marker in the right basal ganglia. Also the plasma CRP. So both the plasma and the CSF CRP were associated with this marker. Just to, as a paraphrase, as an afterthought, we also have shown significant correlation between the plasma and the CSF CRP in these patients. So it's just out there. So then we proceeded to examine how does this impact the neural system. So yeah, I've shown you glutamate, yeah, I've shown you inflammation, but these are all molecules. How do these molecules actually disrupt neural systems? So one of the things is, it turns out that computational studies have shown that there are two types of glutamate signaling. One is called wired transmission. This is a high fidelity transmission that goes from the presynaptic to postsynaptic neurons. It's just one neuron to one neuron, basically. It's nothing more, no complexity here. It promotes synaptogenesis, and it's called wired transmission, or it's just a very high fidelity transmission. The second is called volume transmission, where it's not just one neuron to another, one presynaptic to one postsynaptic. It's one neuron to the next neuron. And what happens is this is caused by these glutamate molecules leaking out into the synapse, and they stimulate neighboring cells. Why? Because you need a tonic activation of an entire region for a function to happen. Remember, none of the brain functions are single neuron studies. I mean, you always go to ACNP or somewhere, and they try to convince you that it's just one neuron just doing all of these things. But actually, it's not. The brain function is complex. It's based on a whole region of neurons getting signaling together. So the problem is, with inflammation, we hypothesize there is a dysfunction, there is an imbalance between this intra and extrasynaptic. In other words, there is an imbalance between the volume and the wired transmission systems. Because remember, inflammation causes more glutamate to leak into the extracellular space. And so this imbalance leads to noise, meaning more extrasynaptic, and decreased intrasynaptic leads to greater noise, chaotic signaling. The signal to noise is lost at that point in time, or it decreases and in the short term. And this often is one of the things you see when people are in acute distress. There are clinicians here probably. You'll see that patients become delirious when they're exposed to cytokines. Even small changes in CRP will show significant confusional symptoms, and this is one of those things, because delirium is essentially a chaotic dysfunctional signaling of multiple neuronal systems. And in the long term, this causes a kind of a glutamate toxicity called excitotoxicity. Now, how do you study this in live human beings? Well, so we went back to functional imaging, and we looked at the literature. And interestingly, when you put a human person inside a scanner, MRI scanner, you see that their brain is not at rest. It's showing some minimal fluctuation. We call this bold fluctuations. It's happening spontaneously. And if you take these bold fluctuations, which is in a time domain, and you convert it into a frequency domain using this conversion called fast Fourier transform, it becomes like a waveform. And then at that waveform, you can study it using measures of amplitude and frequency, etc., etc. So that enables us to study a time series data into amplitude data. And so there are several measures, but what we were interested in is a measure called regional homogeneity. Basically what happens is when one particular voxel, or voxel is the pixel, it's a single unit of the way MR sees the world. When there is a coherence between one voxel and its next voxel, in other words, the concordance in the bold fluctuation between these two systems, the level of concordance matters in regional activation. If they are not concordant, then you have chaos, which is what we were going after. So we were saying that this regional activity basically is a concordance measure of the bold fluctuations of neighboring voxels. And we measured them using this measure called regional homogeneity, or ReHo. And so what we did here was, again, I'm sorry about this complicated slide, but I'm going to quickly make it easy for you. Basically, we took CRP and glutamate, and we developed two groups of patients. We clustered them into two groups, one group with a high CRP and glutamate and one group with low CRP and glutamate. And we measured these ReHo measures in the region where we got the glutamate from and brain-wide. And I'm going to show you some network at this point. So the first thing is, as you can see, there was a significant decrease of this regional homogeneity measure in the place where we acquired the glutamate from, indicating that there is more chaos and chaotic signaling in that part. Compared to the other two measures, the alpha is an amplitude-based measure. It did not show any difference. And the RSFE is a frequency-based measure. So it's not the amplitude or the frequency that changes. It's their coherence between the coherence and fluctuation between one particular set of neurons and the next neighbors. So it's in other words, it's a neighborhood that's not at peace, basically. That's what you're showing. And then we did a whole brain analysis. I don't want to complicate this very much. It's too much of geek technical stuff in this. But nevertheless, the point is very simple. We did a brain-wide ReHo. We got the whole brain ReHo, and then we compared it across these two groups. We developed what is known as the ReHo contrast map. And we found out the regions that were significantly different between these two regions to identify what regions of the brain are misfiring when patients have high levels of CRP and glutamate. And as you can see, we identified 41 different regions that were misfiring. Brain is a big place with a lot of activity. And we then clustered these regions into small groups. And one of the groups, as you can see, subnetwork 4, showed significant structures associated with the reward network, which in turn we felt like was related to anhedonia. So this graph shows you, this is a connectivity matrix. As you can see, this is the low CRP glutamate group, and this is the high CRP glutamate group. On the X and the Y axis are these 41 regions. So it's a correlation matrix, basically. As you can see, in the high CRP glutamate, you can see the loss of the clustering of this connectivity, as you can see. I mean, here it's well-formed, well-connected. Here it's lost. And as you can see, this is a path analysis where we just took the subnetwork 4 that I told you was associated with reward dysfunction. And as you can see, the decreased ReHo in the region, meaning decreased homogeneous fluctuation in the region, gave rise to greater network inefficiency. And we measured that using this graph theory measure called diameter. It's a graph theory measure. And basically it tells you that the network is broad. It's not concise. It's not efficient. It's spread. And so the low ReHo was associated with the broader, wider, inefficient network, which was associated with anhedonia. In the low CRP glutamate group, interestingly, this relationship was lost in the high CRP glutamate group. We believe it was because of the extensive disconnectivity that is seen in these people. So there is no more mathematical association. It's not like something happens, something results. So that whole relationship is lost. I'm going to quickly go through this last because you guys are Jonathan Savage group, so you all know kynurenine. So I'm not going to belabor this very much. Kynurenins are these signaling molecules that mediate, that we believe, Jonathan and I at least strongly believe, mediates the effects of inflammation upon brain, especially. Why? That's because kynurenins are basically metabolites of tryptophan. So in your body, the tryptophan gets converted into serotonin, melatonin, or kynurenine. Of this, kynurenine is the most pervasive pathway. 80%, is that right, Jonathan? 80 to 90% sometimes gets Pardon me? Yeah, it's very good. 95% just goes through this pathway, kynurenins. So the interesting thing about kynurenins is they can transfer and get into the brain, unlike cytokines. And so we all became interested in this at the same time. all of us kind of developed an interest in this. And so one of the interesting thing is these kynurenins go into the brain and start modulating glutamate transmission. That's why we are very interested in this group, at least as of now. And there are two types of modulation. There are two types of byproducts. The kynurenin astrocytes get converted into kynurenic acid, and in the microglia, they get converted to quinolinic acid. So Jonathan has shown some nice data that the ratio between these two, that is the kynurenic acid, quinolinic acid, is of great importance, because this kynurenic acid is a NMDA antagonist. It protects neurons, whereas this quinolinic acid is a toxic molecule. So if this goes down and this goes up, then there is a lot of toxicity in the system. There's some elegant studies that he has published over the time. I would recommend you read Jonathan's 2019 review in molecular psychiatry for that. But we wanted to extend that finding and see if it directly relates to MRS glutamate. And so as you can see, the kynurenins in the plasma are significantly related to, first question was, how is the plasma kynurenin related to CSF kynurenin? And the answer is yes, they are robust, they are dramatically, highly correlated. As you can see, this is the plasma on the x-axis and the CSF on the y-axis. Almost all CSF kynurenins are correlated with plasma. And I think Jonathan wrote like an editorial in the BBI or something recently on the very same issue, that we believe that you really don't need CSF kynurenin measures anymore, because the correlation is so high and dramatic. And this path model is going to show that. Basically here, as you can see, the plasma predicts the CSF kynurenins almost at a 0.01 level, meaning it's extremely significant, as you can see. Whereas, then the next question is, what is this plasma CSF kynurenin association mediated by? Moderated by? Is it controlled by plasma immune or CSF immune? And actually, the path from plasma immune to plasma immune to plasma kynurenin to CSF was so significant. In other words, most of the brain kynurenins is coming from the plasma. It's not coming from the brain sources. It's not coming from other immune sources. And so that becomes a major target of intervention that we can potentially consider going forward. So in order to move this field forward, we just took this, we clustered the TNF alpha and the kynurenins together because they were highly correlated, and developed two clusters, one with the high immune and kynurenin cluster and one with the low immune and kynurenin cluster. And as you can see, that high immune and meaning concurrent elevation of both immune and kynurenin molecules was associated with significant depression severity, anhedonia, and treatment resistance, which was measured using this MGH ATRQ scale. Meaning, in other words, if someone has more inflammation and more kynurenins in their plasma, they're likely to be more depressed, and they're likely to be more anhedonic, and very likely to be treatment resistant. Okay. Here we kind of replicated the same analysis that we did before. So here we asked ourselves the question, does plasma kynurenin, so we basically asked if these plasma kynurenin markers were associated with glutamate changes in the brain, and yes, they were. The kynurenic acid, very much along what Jonathan had reported previously, the increase in the plasma kynurenins were associated with decrease in glutamate, brain glutamate signals, and the increase in QA was quinolinic acid was associated with increased glutamate, and so their ratio was also significantly increased. One point of note here, I just want you to note that this molecule, CSF3-hydroxykynurenin, is one of the byproducts of the kynurenin pathway, and this is a very, very powerful oxidative stress molecule. It's one of the most potent oxidative stress molecules that are found in the brain, and that molecule was significantly associated with astrocytic distress. So I'm kind of presenting a lot of these interventional targets along the way. And this is probably the last couple of slides. Again, we did a network analysis we asked ourselves, what kind of neural networks does kynurenins impact in this analysis? That's what we did in this analysis. And as you can see, most of the neural networks that were associated with the DMPFC, dorsomedial prefrontal cortex. I kind of just spoke with your postdoctoral fellows. The DMPFC is a self-perception region. It's a region of self-perception, negative. It's kind of very significantly impaired impaired in depression. Helen Mayberg did a bunch of studies on that as well. And so this DMPFC to be basal ganglia were the two big nodes that were relevant to this network. And we were able to do path analysis to say that the plasma Chi Neurons directly led to anhedonia by impairing these networks, what we call as the dorsal DMPFC, or dorsomedial prefrontal cortex to basal ganglia connectivity. And these are all the graphs that shows. So thus, these are the final take-home points after about 25 years of career in this. There are like half a dozen points. You can write this in a cocktail napkin, basically. That's pretty much it. That's what happens to what we do. We hum and ha, and eventually it's like 2 sentences. Anyway, inflammation is associated with depression. It may dysregulate glutamate homeostasis, and this dysregulation of glutamate homeostasis is associated with a glial dysfunction most likely, and especially astroglial dysfunction, because astroglia are the ones that process and control glutamate. And when they are sick, glutamate just goes out of control. And the inflammation-associated glutamate leads to disruption in connectivity, functional connectivity and neural connectivity of systems that regulate mood and hedonic functions. And Chi Neuronin is an important mediator in these things. So, I happen to believe, like Jonathan does, but nobody else listens to us. But the fact is, Chi Neuronin may be a better target than blocking inflammation many times, which is what we've been doing. But again, maybe it's enough too, right? So, but these are the final. I want to kind of finish with this slide because this is where we're going to take this to treatment. This is where we're going to try and take this to the patients. This is my last slide. Essentially, as you can see, there are several interventional targets that I've shown you along the pathway. First of all, you can control inflammation using anti-inflammatory treatments, inflammatory blockers, NSAIDs, you can do them. But although I'm not entirely sold on that idea, but I do think there is some thought in that process. We can modify glial. We can strengthen the glial activation. There are several glial modulators that are available. Many of them are being studied in neurology, but nevertheless, they are available for our use as well. There is riluzole, which is actually considered a glutamate regulator, but it's actually a glial stabilizer. Even molecules like lamotrigine tend to do a lot of this glial stabilization business. So I think that's a possibility for us. Microglial modulators are well known. I think you did a study. I think someone did a study on minocycline. You guys did that, right? So there are some microglial modulators available like minocycline, which is a tetracycline antibiotic. Glutamate modulators are well known to all of us right now. Ketamine is out there. Memantine, there's several of those medications that are out there, and several of them are being developed as we talk. This was what I was talking with Salvador. Neuromodulation. Probably we may be able to target these specific circuits, especially DMPFC. And there's these guys from Toronto who've been using that. They're able to somehow do the TMS to target DMPFC regions, and so that's an option for us. Please do not forget this, the oligodendrocyte toxicity, because immune molecules end up hurting oligodendrocytes in a very bad way. And one of the ways of improving oligodendrocyte plasticity, believe it or not, is actually playing video games. It's just been proved in UCSF studies. They've been doing this for a while. You probably know Mesernick. So a lot of these guys, they've been doing this work for some time. It improves oligodendrocyte plasticity. And then senolytics, which is one of those anti-aging groups of medications. There's several of them in development. Many of them are like molecules with the numbers on them. But they're all in development. It's a big market. It's a huge market. It's not being developed for psychiatry. It's been developed for gerontology. I do think at the end, I do want to pay a lot of respect to CBT and alternative methods, because we kind of blown past these things. We're always talking about ketamine, jamming their NMDA receptors. I really think that there is a lot of good things to be said for things like behavioral activation. Robin Appel does a lot of work on that. There's a lot of good things to be said about those. And those things work better than many of the medications that I prescribe day in and day out with far less side effects. And in fact, in the process, they actually improve their health as well. So TMS, all these things are very useful things. I do think that there is a lot of potential for that. And also at the Emory now, we've just started using the ultrasound. One of those Georgia Tech guys, they've developed some kind of an ultrasound wave that can, they've been using it for Parkinson's, I think. They're trying to do it almost like DBS for subthalamic body stimulation. So anyway, some of these, there are several targets that are available along this way. I want to finally finish by thanking my collaborators, Andy Miller, we all know him, at least many of us know him quite well, and Jennifer Felger. Senchuan Chen is my imaging spectroscopy